We're well into our second season of OMG OMx, and thanks for continuing to come back to hear from all of our speakers. Hi, Bjorn, and I wanted to welcome you to OMG OMx and thank you for appearing on the podcast. Perhaps to start with, you could tell us a little bit about your story, since you do work for a mass spectrometry-based company, but you started in a very different place.
Yeah, of course. Yeah. Thank you very much for having me. It's a pleasure. So I started my career with a very theoretical orientation. I'm a mathematician by training, and what I did back then was math for math's sake, with almost no real life application. So if you would be interested, it would be about triangle group operations on Riemann surfaces. And the topic was located somewhere between like higher algebra, topology, and complex number theory. So it was very, very, very theoretical. And I realized back then that I would like to do something with a more impact on the problems of the real world. And I then found a PhD position with Achim Tresch at the Gene Center in Munich, which belongs to the LMU here. Achim back then was looking for someone that had a solid understanding of statistics, as he himself also did study mathematics. And this turned out to be a very, very interesting position for me back then. So I had to kind of learn a lot of the biology and biochemistry from scratch. And I could apply all my mathematical and my statistical knowledges in formulating models. So back then we developed a model to estimate the half-life of mRNA transcripts, which turned out to be actually a quite cool thing in the end. It worked quite well. After that, then I joined Patrick Kramer's group as a postdoc, also at the Gene Center. He was back then heading the Gene Center. Now he is heading the Max Planck Society. And I also then moved with him to the Max Planck Institute in Göttingen as a project leader. There we did a lot of multi-omics approaches, all NGS based, to understand the dynamics and checkpoints of the enzyme that transcribes messenger RNAs. So then we develop models, for example, how fast the elongation velocity is of the polymerase, and what are the typical things that the polymerase in different steps undergoes during transcription, how these phase transitions is, how the termination is, and so forth. And then roughly 4 years ago, I then switched completely from academia to industry. And I joined, and also from transcriptomics to proteomics by joining a CRO called Evotech, where I first got into touch with mass spectrometry. I mean, I've heard about it. I have heard talks about it. I used a little bit of the data back then for a correlation analysis when it fit the biological topic we had back then. But I did not analyze these data myself to that time. And that happened at Evotech, that I completely switched gears. And then roughly 2 years ago, I then joined NEOsphere Biotechnologies, heading the biostatistics department. And that's where I am now.
That's really fascinating. And I kind of want to ask you an off the cuff question on some of the modeling that you did in your training and career. Have those found practical implications, or is it the statistics and the modeling and the energy distribution just for the sake of understanding that?
Yeah, so a lot of things you do in science first is for the sake of understanding. But in this case, for example, when it comes to RNA half-life, is actually a very valuable information, especially nowadays when we have RNA-based medicine, because you'd like to formulate them in such a way that they are very stable and have a lot of translational potential, right? And so there's a lot to be learned from what makes an RNA stable. and what things, what modifications, and what aspects of it can you mimic in your drug, for example. So that's why I always thought it would be like it was actually a cool topic. It started out very much for the science sake, understanding much more of how the cell works and so forth, but it turned out to have some implication later. Yeah.
Do you see similar things at Neosphere, since you're, again, you're parallel to the science, but you're doing biostatistics? So is there anything going on there that you think is relatable?
Here it's even more applicable, right? We since we actually do screen or characterize all types of degrader molecules, it's basically we try to find as many starting points for drug development as we can, and that is a huge application.
That's great. You told us a little bit about some of the people that have influenced you along your journey. Is there anybody else, maybe even again from your more recent relocations into industry?
Yeah, that's actually a good question. So I would say the major influences I had actually when I stayed in my academic career. I actually there are a lot of people I work with at Evotech as well as here, which I learn a lot from all the time, which is very valuable. Yeah, but a few crucial things that I think kind of endured over time as valuable things are actually from my early days.
How did you get interested in science or math in the beginning? Is that something that's been with you since your early childhood or early days?
Yeah, so I back then I did not really know what I wanted to study initially, but I always liked math, but I was never that good at it that I thought as of it as a career choice. I then started studying something else, and actually there was a part math that needs to be done for the studies, and I realized that I like that above all, and so I decided to actually switch gears and study pure mathematics at that point, and I never regretted it. I still love mathematics. It still fascinates me, and I'm glad I did that.
Yeah, thank you for sharing that. So maybe we can go back to where you are now at Neosphere. What are the goals of this company, and what it makes it so attractive to you to be part of this?
So we are a biotech startup in the south of Munich, and our focus is targeted protein degradation, or short TPD. So TPD utilizes the waste disposal system of the cells to selectively eliminate disease-causing proteins. And as such, it is a promising alternative to existing therapeutic modalities. And we have established here a deep proteomic screening platform to characterize all sorts of degrader molecules. And we actually believe that only proteomics using mass spectrometry at that scale and depth and quality that we are able to deliver can reveal the true target scope of TPD.
And then what else? I think in looking a little bit about Neosphere, I saw things around high throughput screening and automation. Is that something that you have any interaction with?
Yes, so scalability and automation are two very important aspects for us. It is our ambition to be able to screen large degrader libraries and guarantee fast turnaround times. So we developed dedicated web apps that make almost every process intuitive and easy to use for all our colleagues. This ranges from pre-acquisition workflow automation, intra-acquisition live QC monitoring, post-acquisition server job submission and control, as well as visualization tools for assessment, review, meta-analysis, and all the way to the biological relevance of the findings. So everything should be very seamlessly and be able to retrieve with a click. So that also all non-bioinformaticians are very capable of doing this on a daily basis.
How do you feel about supporting a system that requires sample preparation, instrumentation, when this isn't something that you've spent most of your time learning about and doing?
I find it very exciting, to be honest. So it is an additional challenge to also cater to these aspects of what I would call upstream work. For example, how can these processes be tracked? How can the nomenclature of biological material and methods be controlled? And I actually have quite some good grasp of some of these processes, as I also done some cell culture work in my past. So I have a little bit of more connection to it than other bioinformaticians might have.
And then how does this become helpful for you to do data analysis? Do you think doing that day to day or having some understanding is important for you to be able to do your job, to do the complement?
Yes. So I feel that you get a certain sense of biological and technical variation, and this helps you to appreciate even further that statistics is giving you probabilities and likelihoods and not actual facts. And so, especially for me as a mathematician, this was actually a hard thing to understand because in math, it's logical. It's either that way or the other way, right? So as soon as you enter the realm of noisiness that you have in biological systems, it's a very different approach to thinking about it.
That's really quite interesting because having done, basic statistics and a lot of biology, I'm not sure I ever would have made that connection. So, yeah, the noise of biology is a really great phrase, I think. Okay, so then let's talk a little bit about how you do development and processes at NEOsphere. How much of it is truly intentional, and how much of it is you stumbling into it to try and address something that's come up on the biological side?
So in our case, most of it, I would say, is intentional, but there is always a certain part that you stumble into, especially when it comes to new exciting data formats or programming packages. And such develops can then suddenly lead to major improvements, right? And if you have kept a clean code base, then these things are also easy to integrate, but might give you like a real edge. And these things are often things you stumble upon reading blog entries or news about certain platforms. So I always compare programming larger data analysis pipelines to building a house, like a house of cards. So you need to make sure that your foundation is solid because otherwise it might collapse at some point. And you're constantly busy fixing problems instead of extending the functionality, right? So that's so if you have a lot of intention in the beginning, you know where you want to go and you know which base you need to lay down, right? So that's why it's actually good that it's not that much stumbling into, because that might actually then be not that easy to integrate anymore. But there's often, or there are some parts where you stumble into, yeah.
Bjorn, can you tell us a little bit more about how you best serve your customers and what technology you offer to them? I think you mentioned an app before when we chatted.
Yeah, sure. So I cannot claim that I understand the biology as well as my colleagues and collaborators and customers, but I can give them the tools to understand it even further. Right? Visualization of data is can be a very powerful tool, and we do not just use the platform we built, the platform of web apps internally, but we also offer this platform to our collaborators and customers. And so far, we've received a lot of positive feedback over the last time. Given that a lot of the workings in bigger companies often is making plots in Excel files and so forth, and they love the idea to have a very easy way of clicking somewhere and be hovering in an interactive plot and reading all kinds of data to the depth that you would never reach in an easy format.
So where do you see NEOsphere going next? If that is something you can share, do you have any big projects or big, next milestones that you're trying to achieve?
TPD itself is a very hot topic, and it will become even bigger in the future. And I think we will be a big part of that.
That's great to hear. So my last question for you, since we're recording this during the Olympics, I wanted to ask you if you think you're more of a mathlete or an athlete. And if you pick the second one, what would you compete in at the Olympics if you could do anything?
I would actually say that I'm more of an athlete. I'm a very sporty person. I like to ride my bike to work. I do a lot of bouldering, which is
Okay.
like climbing,
Rock
but
climbing. Yeah.
without the rope. So what would I compete in at the Olympics? That that's actually a very good question. There there are a lot of disciplines. Hmm. I'm actually not sure if climbing is an Olympic discipline.
There are some offshoots of the Olympics that do like extreme sports, and that might be one there. As we end today, is there anything else that you'd like to leave us with, or any fun facts that you'd like to share with the listeners?
Yes, sure, of course. Back then, when I was still in my academic career, I actually found a Nature article about color blindness and the importance of the choice of colors back then. And I and then I got very much interested in this topic. So it basically calculated the probability of given like where your reviewers are located, what the probability would be that one of your reviewers would be a colorblind individual, and you need to take care of your figures in order to have a better chances. And I got very much interested in the topic, or especially in how to enhance digital pictures for colorblind individuals. And I basically looked up the existing algorithms back then, and I had an idea of how to improve it. And I actually then wrote a small algorithm to do that, which I then applied to artworks, for example, such as the Mona Lisa. And the results were actually quite funny. I never had the chance, however, to test it on a colorblind individual and if it would actually have an improvement in the perception. But they looked actually quite funny.
I think you've got a backup career there if you ever need one to, partner with some other scientists and investigate that. All right. Well, thank you so much for being on OMG OMx. It's always great to hear and learn from people out in the field. So I appreciate your time.
Thank you very much. Yeah, it was a pleasure being here.
The merging of different academic disciplines isn't always obvious, but I hope that Bjorn was able to show you today how much math has an influence on biology and other systems. Thanks for listening and staying with us today.